Hello, everyone. Welcome to the second lecture of MD 6028. Today, we're going to be talking about virus classification. So last week, hopefully, you gained a good, strong understanding of what viruses are, how we define viruses, what we're actually talking about when we're talking about viruses, kind of how viruses are different from bacteria, and all these fundamental ideas that sit at the base of virology. A lot of what we talked about last week, you may have heard in bits and pieces in various places. Hopefully that lecture brought it all together and gave you this solid foundation. What I want to do this week is build upon that foundation and talk about one of the key fundamental aspects of virology, which is classification. So, as we said last week, we talked about defining viruses. Now we need to talk about understanding the differences between viruses. If you like, last week we talked about what all viruses have in common. What are these overarching characteristics by which we define viruses? This week we're going to look at viruses and say, okay, how can we split these viruses up into groups? How can we classify the different viruses? Obviously, we know common cold is very, very different to poliovirus. Or I should say the common cold is very different to polio, and the reason is because the causative viruses are different. So the rhinoviruses that cause the common cold are very different to polioviruses that causes polio. They're very different in turn to the herpes viruses that cause chickenpox and herpes simplex. So what are the differences? That's what we're going to understand today. We're not going to go into pathogenesis, so we're not going to go that far down the road. We're just going to look at the different groups that exist. So we're going to look at the different types of viruses we have, and then we're going to start to understand why different viruses are put into these different groups. So the first thing to understand is this idea of classification. So today we're talking about the classification of viruses and how we can group viruses into different groups. But we don't just classify viruses, we classify all forms of life. So this shouldn't be a new concept to you. It's fundamental in biology. It shouldn't be a new concept to anyone, even if they've never studied biology. We know that we group forms of life together based upon similarity. We have reptiles, for example, we have mammals, and we have fish. These are three groups of organisms that we've grouped together based upon their similarities. So reptiles, all reptiles have common shared features. That's why they're classified as reptiles. All mammals have common shared features. That's why they're classified as mammals. All fish have shared features, common shared features. That's why they're classified as fish. This classification that we use to come up with these groups is called phylogenetic classification. So the intuitive groupings that we know that I just talked about, these are actually based upon phylogenetics. And phylogenetic classification is essentially classification based upon genetic similarity. And when you classify organisms based upon genetic similarity, you end up with a tree of life, because the key thing to remember is that all organisms on Earth descended from a single common ancestor. We call this common ancestor the LUCA, the last universal common ancestor. And the fact that there is a LUCA, the fact that all organisms on Earth are descended from this single microorganism, means that if we map all organisms based upon genetic similarity, as I said, we will have this big tree of life, and at the base of that tree we will have LUCA. So as we move up through the tree, through the branches, up towards the leaves, we're getting further and further away. So organisms are diverging, types of life are getting more and more different. However, right at the base of that tree, you know, a long time ago, 4 billion years ago, is it 4 billion? Yeah, about 4 billion years ago, we have the last universal common ancestor. After the last universal common ancestor, organisms started to diverge. First, we have the bacteria and archaea and eukaryotes. Then there was further divergence, so the eukaryotes eventually diverged into fungi and plants and animals, and then there's further divergence of the animals into the types we've talked about, mammals, and then eventually we end up with all these different species, and that's how the phylogenetics works. It's mapping out this massive tree of life based upon genetic similarity with Luca at the bottom. So it's really important we understand what phylogenetics is, and the fundamentals is it's the way we classify organisms, macroorganisms and microorganisms. So whether you're talking about a big giraffe or a little bacteria, it doesn't matter. You can classify them phylogenetically because they all descended from the last universal common ancestor. And again, this classification is based upon genetic similarity. And what we see is that the closer organisms are genetically, the closer they are on the tree. And so we end up with these branches on the tree of similar organisms. So groups of organisms, again, reptiles, fish, mammals, etc., etc. This is useful. It's inherently interesting because it's interesting to know how all these organisms originated. It's interesting to know their evolutionary path, but it's also inherently useful because if I tell you an organism is a mammal, if it belongs to this group called this branch called the mammals, you already know a lot of information about it because you know it shares some key features with the other mammals. Same for reptiles, same for fish. When we're talking about microorganisms, this is super important because microorganisms cause infectious disease. So if we can classify microorganisms phylogenetically, and straight away we can say, oh, it belongs to this group, or it belongs to this group, or it belongs to this group, we can straight away say, okay, maybe this treatment will work, maybe this therapy will work. We're saying this microorganism is similar to other microorganisms, so why don't we try the same therapy? And the most simplistic way to demonstrate this is by talking about antibiotics. So we know antibiotics are used to treat bacterial infections. Antibiotics work against bacteria. So if we take a microorganism that's causing an infectious disease and we define this microorganism as being a bacteria, so we're putting it in that phylogenetic group, because if you remember, bacteria is this huge branch of the tree of life that separated from other branches quite early on near the bottom, so near Luca. If we put our infectious agent in that group, then we can say, okay, this is clearly a bacteria, so we can try to treat it with antibiotics. So hopefully that's a clear example of how grouping organisms together is useful. If we want to go into even more detail, we can look at, what's a good example? Mycobacterium is a good example. So Mycobacterium is a family of bacteria. So it's a grouping within the big group of bacteria. So if you can imagine this big branch of bacteria, then there's a smaller branch called Mycobacteria within the bacteria. It's similar to how, we have this big branch called mammals, and then within this we have little branches, like we have the dogs and the cats, that kind of idea. So going back to the Mycobacterium example, we know two microorganisms belong to the Mycobacterium family that cause human disease. We have Mycobacterium leprae that causes leprosy, and we have Mycobacterium tuberculosis that causes TB. These two infections can often be treated with the same type of drugs. We call those type of drugs antimycobacterials. So they're a subclass of antibiotics that specifically work against mycobacteria. So again, just by knowing these diseases caused by these two infectious agents are caused by bacteria that belong to the same little subgroup, we know, or we have an idea, that potentially the same small group of drugs can be used. So this classification is really, really important. Classification really does matter. Just some technical terms here. You probably already know this, but it is important to understand what these terms are. So when we're talking about the tree of life, we have LUCA right at the bottom, then we have the tree starts to branch off and split off. And as it splits into these more specific groups, and these are essentially the names of the different groups. So as you would expect, as we go up through the tree, the number of groups increases. So there's a huge number of different species of organism, but there's only 3 domains. Literally only 3 domains include all types of life. So we have archaea, and then we have the group bacteria basically, and then we have eukarya, which contains all the eukaryotes. So these are the first 3 branches of the tree of life. Then it continues to branch into kingdom and phylum, class, and then orders, then families, then genus. Then eventually we get to species, which is the most specific classification. So yeah, these are fundamental biological concepts you'll probably be aware of, but if not, just make sure you're super clear on what these different classifications mean. You don't need to memorize them, you don't need to memorize the order, just understand the concept, understand why there's only 3 domains, but there's hundreds of millions, or however many there is, different species. It's because we're thinking about a tree of life, and species is right at the edge of the branches. Here's an example. So I've just got a couple of images here. They are pulled from Wikipedia, but that's absolutely fine because I'm trying to make the point that this is well-known information, that it's really fundamental, that you can get it anywhere. You can literally see we have the species. So we have giraffe as the example here. Species, then genus, family, order, class, phylum, kingdom. We don't have the domain because it's assumed we'll know the domain. It's not a bacteria and it's not an archaea. It's obviously a eukarya, or a member of the domain eukarya. Same for humans here. We have Homo sapiens, and we see that we are in the same phyla as giraffe. We see we're even in the same class as giraffe because we're mammals. So we might think of ourselves as pretty distinct from giraffes in many ways, and in many ways we are, but we are alongside them on the tree of life up until a certain point. Because if we compare ourselves to giraffes, we do have a lot of shared features, and we have all the shared features that mammals have. If we compare ourselves to bacteria, which are very far from us on the tree of life, they're in a different domain. We don't have much in common with bacteria at all. They're single-celled, prokaryotic microorganisms. And so they're far away from us on the tree of life, and that's pretty obvious. We can see that. Okay, here's an example of a bacteria, and the example of a bacteria of a bacterial position in the tree of life. We see the domain here is bacteria, so it's not part of eukarya like us. It's part of bacteria. So again, this is a single-celled microorganism that is prokaryotic. And yeah, we just have the different classifications here. You'll see the actual groups it belongs to, completely different to the ones we belong to, completely different to the ones the giraffes belong to, but the names of the levels are the same. So we still have a domain, and we have a phylum, and we have a class, we have an order and a family and a genus and a species. So the same system is being used. It's just the specific groups that the bacteria are in are different. So it should be clear why this classification, this phylogenetic classification, is used, because it's such an all-encompassing system. It's such a powerful system that it can include all forms of life on Earth, even forms that exist that are as vastly different as they are. It doesn't matter how different we are from bacteria, we still fit on the tree of life together. We might be very far apart on the tree, but we're still on the same tree, and because we all descend from the last universal common ancestor. Okay, and here's the problem. Viruses don't fit on the tree of life, so everything I just said doesn't actually apply to viruses. It's a difficult one, a very difficult one. All organisms are descended from Luca, as I've said, except viruses. Viruses aren't descended from Luca. We don't know where they're descended from. That's a problem because we can't classify viruses phylogenetically. We can't fit them on the tree of life because they're not descended from Luca. They're not part of any of the 3 domains of life. We map the genomes of different organisms to fit them on this tree of life, as I said, and then we slot them on where they're most similar to other things. We're kind of mapping this genetic similarity. The problem with viruses, when we try to do that, they don't fit anywhere. It's impossible to figure out where they evolved from. They don't appear to have evolved from anything on the tree of life. They just don't have a place there. No one's exactly sure how they arose or where they arose, or even how many times they arose. So there's this idea of viruses as being polyphyletic. So if on the tree of life, generally we're looking at monophyletic organisms. So for example, mammals, there's one group of mammals. Mammals evolved once, and that's the group of mammals. They evolved from this branch, and that's where they are. That's the idea of being monophyletic, the idea that all reptiles originated at the same point. So the branch of reptiles only spawns off once. Doesn't appear to be true for viruses. In fact, it looks like viruses may have arisen separately at least 7 times. So what that means is not only are viruses not related to the organisms on the tree of life in any way that we can tell, they're not even related to each other. So if we try and organize viruses into trees of life, we manage to split them up into 7 different trees, and these trees don't appear to be related to each other. So what it looks like is that viruses arose 7 different times. So they're polyphyletic, poly meaning many. Quite confusing. It's just the way it is. That's all I can say there. And it's actually further complicated by the fact that even when we arrange viruses into these separate trees, most viruses don't fit onto those trees. So most viruses don't even appear to be related to these 7 separate trees that we've 7 or 9, depending on the literature you look at least 7. Viruses don't appear to be, most viruses don't even appear to fit on these trees. So it's difficult enough that we can't fit viruses on the tree of life. Not only that, we can't fit viruses onto their own tree of life. They have to be assorted into at least 7 different trees. 7 different trees that don't appear to be related to each other. And not only that, but most viruses don't even fit onto those 7 trees. So we just have no idea how they're related to the different types of viruses that we have. Absolute chaos in terms of classification. There have been efforts in recent years to classify viruses in a slightly different way, which we'll talk about. Just an effort to update the classification system that is used. But yeah, in terms of this classical phylogenetic classification, it really doesn't work for viruses. It's problematic. Those trees of life that we do have for viruses are stunted. So they're very, very short trees. They're not these big branching. Sorry, they're not like the big branching tree we have as a tree of life, which goes all the way down to domains until last universal common ancestor. They're just short trees that only contain species, genus, family, and order. So we can essentially group some viruses into these different orders. And because we can't group them all the way down to domain, we call it a stunted tree. It's quite a small tree. But yeah, we can classify them into these between 7 and 9 different orders, and that's the best we can do. And each of those little trees I talked about represents a different order. So we have the order and the family and the genus and the species, but we can't relate these trees to each other. We can't group them together into any classes or phyla or kingdom. But yeah, it's the best we can do right now. So if you look up the classifications that we do have for the different viruses, you'll see that they don't go higher than order. We just don't have a class or phylum or a kingdom in the systems that are currently widely used. So for example, we have HIV here. We have the species, genus, family, subfamily, which is subdivision of family, then family, then order, then nothing else. Don't have a class, don't have a phyla, don't have a kingdom, don't have a domain, because we don't know. We just don't have those answers. Same for Ebola virus. So we have the different species of Ebola virus here. We also have the genus and the family. Then we have the order. Then we have no class, no phyla, no kingdom, no domain. Again, because we just don't know that information. We're just not able to classify viruses at that level yet. Or I shouldn't say yet, we're just not able to. Hepatitis B virus, species, genus, family, fine. Under order, we have unassigned. So this is just to demonstrate the point that I was making earlier, that most of the viruses that we know about, we can't fit into the orders that we have. So we have those between 7 and 9 orders of viruses. So those 7 to 9 little trees of life that we talked about, that only include viruses. However, most viruses can't even be slotted onto these trees. Most viruses are just, we just don't know how they're related to these trees of viruses. HBV is an example of one of these viruses that we can't really understand how it's related to other viruses or to other viruses on the trees. That's why it's listed as unassigned under order. And the importance of HBV as an example is that HBV is an inherently important virus because it causes a lot of human disease. It causes liver cirrhosis in a lot of infected people. It's quite serious. It's quite chronic. It's an issue, and there are a lot of people researching hepatitis B virus. However, despite this, we don't really have an understanding of how it is related in an evolutionary sense to most other viruses. So don't think that all the viruses that don't have orders assigned, all the viruses that don't fit on these mini trees of life are just unimportant, or they just haven't been studied, because that's not the case. Actually, some very, very important viruses, like hepatitis B virus, that we are very interested in, have not been assigned a position through phylogenetics in our wider understanding of viral classification and relatedness. So we don't actually understand how some of these very important viruses are related to most other viruses. Despite the fact we can't classify most viruses into an order, we can classify most viruses into a family. So even the viruses we can't classify into an order, we can usually classify into a family. And a family is essentially just a big group of related species. So we can take a bunch of different virus species, we can decide these are sufficiently similar to each other to be classified as a family, and then we can name that family, and that's essentially how we come up with these families. We also have genus classification, so we will further group these related species into a genus, and we'll have multiple that fit into each family. The example here is HIV, and the family to which HIV belongs, Retroviridae. So Retroviridae is the family name. We have a bunch of different genuses listed here: alpha retrovirus, beta retrovirus, gamma retrovirus, and these are all members of the Retroviridae family. Within each genus, we have the species that fit within that genus, and again you see we're grouping these viruses based upon similarity, and that similarity is genetic similarity. So we are using the phylogenetic system still. We've just accepted the fact that we can't group viruses higher than order using phylogenetics. We just deal with it. We've accepted the fact that most viruses can't even be classed into an order. We just deal with it. But what we can do is group viruses into families at least. So again, we are using the phylogenetic system here. We are grouping the viruses based upon genetic similarity. So yep, the example here is Retroviridae. When you see this suffix viridae, you know we're talking about a virus family. And I did mention this in the lecture last week. The other example I gave was Filoviridae. That's another family. Also Poxviridae, Herpes viridae. These are distinct families of viruses, and within each of these families we have a range of different virus species that are related to each other. Okay, and just a bit more information here on virus families. So it is a useful classification. Again, I talked about one of the reasons why phylogenetic classification is useful is because it enables us to design therapeutic interventions. This is still true for viruses. The example I gave earlier was bacteria, but it's true for viruses. If we think of the retroviruses again, so Retroviridae is the family, antiretroviral drugs will be useful to some degree against most members of that family. So if you design a drug to target HIV-1, which is a member of the Retroviridae virus family, that drug will likely have at least some activity against HIV-2. So it is useful to classify viruses into families. It is useful to use this phylogenetic classification, even if it has its limitations. What else do I want to say here? Okay, there is a point here that I need to make about species classifications. So we tend to think of a species as being a very distinct thing. This is this species, this is this species, and this is this species, and these are separate things. It's a little bit more complicated when we're talking about viruses. Even though we do have distinct species of viruses, you know, HIV for one, and all the other different examples, it's not quite as simple as that, because each species is actually a spectrum of genomes. So because viruses mutate so fast, because they produce so many progeny, all viruses belong to a single species, so all members of that species are not identical. Sometimes they're not even, well, they're similar, but there's questions over when they become a new species. When do we actually have to put a line down the middle and say, okay, these members of the species are just too different from these members of ostensibly the same species, but the difference is so great that we actually have to split them. HIV-1 and HIV-2, for example, two human immunodeficiency viruses, but they're sufficiently different that we classify them as two separate species. So just bear in mind that this idea of species, it does start to break down just a little bit when we're thinking about viruses, because they replicate so fast, because they produce so many progeny. So just bear that in mind. Okay, so hopefully I've convinced you that we can kind of classify viruses phylogenetically, but it's not a perfect system. It doesn't work as well for viruses as it does for other microorganisms and other organisms. So is there a better way? Is there a better way we can classify viruses? Is there a more effective way we can group them? And the answer is yes. We use phenotypic classification. Phenotypic classification is a very different classification system that works for viruses. So all the issues we have with phylogenetic classification are essentially avoided when we use phenotypic classification. When we're talking about phenotypic classification, the clue is kind of in the name, phenotype. So we know what phenotype is. Phenotype is what's observable, so the observable characteristics of something. In terms of viruses, we can literally pinpoint what phenotypic characteristics they have and use that classification. And use those features to classify the viruses. So let's think about what a virus is. Let's think about what phenotypic characteristics we can observe. First off, we said that viral genomes are very different to our own genomes, and they're also different across different viruses. So all our cells have double-stranded DNA genomes. Viruses have very, very different genomes. Viral genomes can be composed of DNA or RNA. The genome can be single-stranded or double-stranded. And if the genome is single-stranded, it can be positive sense or negative sense. And we're going to talk about what positive or negative sense means when we talk about viral replication specifically. But for now, the best way to think about it is if you have our genome, so our double-stranded DNA genome, you have two complementary strands, but they're not exactly the same. One is positive, one is negative. They're complementary, but they're not exactly the same. The viral genome can be in a positive strand or a negative strand. And that implicates how the virus replicates, essentially. And again, I know I'm being quite vague, but it's because we're going to go through all this in detail down the line. All we really need to understand at this stage is that the viral genomes come in different forms, and we can classify a virus dependent upon what form its genome is in. So whether a virus has a DNA genome or an RNA one, or a single-stranded one or a double-stranded. So you can have a virus with an RNA genome that's single-stranded and that is positive. And this classification, firstly, it works. So all viruses will fit onto this classification here. Secondly, it's useful. So you can get a lot of information about a virus by knowing what its genome is composed of, by knowing if it has a DNA or RNA genome, by knowing if its genome is single-stranded or double-stranded. So it's a very useful classification system that works. Another characteristic we can use is whether or not viruses have an envelope. So we've mentioned in the past that some viruses have an envelope and some don't. This is useful in terms of classification because we can take any virus, we can say it does have an envelope or it doesn't, or we can put it into one of these two groups. So all viruses can be classified in this way, just easy, whether or not they have an envelope. And it's also useful, again, in the practical sense, because it gives us information about the virus. Whether or not a virus has an envelope has significant implications on its pathogenesis, on how it infects cells. So it's useful information. There is a temptation to kind of think, okay, we'll classify viruses based on whether or not they have an envelope. But why? Who cares? What difference does it make? The difference is real and it's significant because it tells us a lot about how they replicate. Same for all the information about the genome I was talking about. Whether or not the genome is DNA or RNA, as I said, it has a significant impact on the way the virus replicates. So it's a useful classification system. This is what we end up with when we apply this classification system. And unlike the phylogenetic classification we talked about, all viruses can fit onto this graphic I've got here. Obviously, I don't have every virus family listed, but if I were to take any random virus family and put it on here, it would have a place. You'll notice I'm saying virus families because the names at the bottom, these are not individual viruses, these are virus families. And these families are derived from phylogenetics. So what we're doing is we're stealing a little bit of phylogenetics. So we're using phylogenetic principles to group viruses into families, and then we're placing those families onto these phenotypic maps. So the overall mapping here is based on phenotypic classification, and you can see that by comparing this to the last slide we looked at. So you can see on the left we have DNA viruses, on the right we have RNA viruses. Then we split up the viruses dependent upon whether they have double-stranded DNA, DS, or single-stranded DNA, SS. For the RNA viruses, we then use the further classification of positive or negative. So the single-stranded RNA viruses, as you can see on the right, can be positive, SS plus, or SS minus, negative. So they're classified by this characteristic as well. Finally, we have the envelope or unenveloped classification. So we can say, okay, this virus is, for example, an RNA virus. It's a single-stranded RNA virus, and that single strand, so that single-stranded genome, is positive sense. Is it enveloped or unenveloped? Does the virus have an envelope or does it not? If you know, you'll see some examples here at the bottom where we have an RNA virus that is single-stranded and it's positive sense, and it's enveloped. HIV is an example, retroviruses. And then in the unenveloped camp, we have the cliquey viruses, so cliquey viridae. So this classification works. It's really useful. Once you understand more about viruses and the way they operate and the implications of these characteristics, you'll understand just how important this is. Once you have the information in your head, you can kind of take a step back and you can see almost patterns. You can see, oh, these RNA viruses all have this in common. These single-stranded RNA viruses all have this in common. Oh, there's this reason why there's only one family of single-stranded DNA viruses. So it is really useful. It's such a helpful system for classifying viruses. I hope I outlined in the last lecture just what a nightmare it is to classify viruses phylogenetically. It's just chaos. It really doesn't work very well. But this classification system does work very well, and it's widely used. So just a little bit more information on the classification characteristics that I introduced a few slides ago. So first off, we can look at the genome. So we said genome can be composed of RNA or DNA, and it can be single-stranded or double-stranded. Okay, so when we do this classification, when we apply this classification, we split viruses down the middle. So we say these viruses are DNA viruses, these viruses are RNA viruses. Very, very distinct groups. They also have distinct features in terms of the DNA viruses together. Because they have DNA genome, they have certain things in common that are associated with a DNA genome. Same for the RNA viruses. Because they have an RNA genome, they have certain features in common that are associated with an RNA genome. One thing you'll notice straight away looking at these is that there are not many single-stranded DNA viruses, and there are not many double-stranded RNA viruses, or virus families, I should say. There's a reason for that. The reason is DNA is most stable when it's double-stranded. So most viruses that have evolved to have a DNA genome have evolved to have a double-stranded DNA genome because single-stranded DNA just isn't very stable. The reverse is true for the RNA viruses. So RNA is most stable when it's single-stranded. Double-stranded RNA is evolutionary. It's quite rare. It's just kind of tucked away on the end of the diagram there. Most RNA viruses are single-stranded, again, because it's the most stable conformation for RNA. So straight away we're getting information from this classification system, and hopefully we're starting to see some of the reasons why it works. I kind of make a vague statement in this slide here where I just say the implications of genome composition in terms of whether the virus is RNA or DNA are significant, and they're quite wide-ranging. And this is absolutely true. It's not the right place to go into detail on this because, again, we do go into detail regarding genomics in later slides. It's really, really interesting. It's complex, but when you wrap your head around it, it just makes sense, and it just feels so intuitive. So when we first approach it, it'll seem complex, but as you work through it, it all comes together. For example, you'll notice a lot of these RNA viruses are almost quite dangerous viruses, viruses that spread quite rapidly, viruses that are responsible for a lot of epidemics and pandemics. Coronavirus is an RNA virus. It's not a coincidence. These RNA viruses, again, they tend to be single-stranded, as we said. And one of the consequences of being single-stranded is you have a very, very, very rapid mutation rate. We'll look at exactly why, but that's kind of an example of how these patterns exist. And when we apply our understanding of virology to this phenotypic classification, it really helps us to pull these patterns together and get a really kind of comprehensive, wide-ranging and quite nuanced understanding of virology. So we can also classify genomes, as well as saying that DNA or RNA, we can classify them as single-stranded and double-stranded, and the single-stranded genomes can be classified as positive or negative. We will often use the term positive or negative sense. And this is a diagram that should help to explain that. So if we think about, and I know I mentioned this briefly earlier, but this is a bit more in depth. So if we think about our genome, we have double-stranded DNA. As I said, those two strands are not identical. They're complementary, they pair, they base pair, but they're not identical. One of those strands is a template strand. The other strand is an encoding strand. So the encoding strand has the information, has the order, the base order that will be translated or transcribed into RNA and then will be translated into an amino acid sequence. The template strand is just the opposite of that. So it's just, or I shouldn't say the opposite, it's the complementary of that. So it's the strand that is binding to it, it's base pairing to it, but the template strand itself doesn't actually encode the information. It's the encoding strand. And this is essentially the difference between positive and negative. Positive is just another way of saying encoding. So the positive strand is encoding and the negative strand is the template. When we're saying positive and negative, we're applying these ideas of encoding and template strands to the viral genome. So if we have a single-stranded genome, which we do for the single-stranded RNA viruses that we're talking about, that strand can be positive or negative. That means it can be template or encoding. So this strand can either directly be carrying the information that determines the amino acid order, or it can be complementary. So if you have a negative strand, of RNA, so a negative stranded genome, that can't be directly used to produce protein because it doesn't contain the information. It first needs to be copied into a positive strand. A positive strand contains the right information. Hopefully that makes sense. I know it's a little bit of a tricky concept. We'll talk about it in the tutorial because it's one of the many concepts that when you first look at it, it looks really tricky. But then when you pull it apart, when you get to grips with the definitions, it all makes a lot of sense. Okay, and here's kind of a bit of exposition around that. We have two strands of RNA here. The top one is positive and the bottom one is negative, and then just kind of demonstrating the idea that these are two halves. So we have positive and a negative. They're complementary. They're not exactly the same. They're complementary. And if you have a single-stranded virus, again, most single-stranded viruses are RNA, you're just going to have one of these. You're going to have a positive sense genome or a negative sense genome. If it's a positive sense genome, like at the top, it can be turned straight into protein. Because remember, these viral genomes are RNA, so when it comes into the cell, if it has a positive sense genome, that's positive RNA. That's essentially mRNA. That can just be translated by host proteins, so translated by cellular machinery and turned straight into viral proteins. For negative stranded genomes, so for viruses with negative stranded RNA genomes, it's a bit more complicated because they have to be first turned in to positive RNA, then that positive RNA can be translated by host cell machinery. And that's another key thing to remember, that when we have a virus with an RNA genome, when it comes into the cell, it doesn't need to transcribe its genome. Its genome is RNA, so it just goes straight for the process of translation, hypothetically. It has an RNA genome, so if that RNA genome is the right type, if it's positive, if it encodes the information, it can be translated by host cell machinery straight into viral proteins. A consequence of this is that positive stranded RNA viruses replicate in a very, very different way to negative stranded RNA viruses. So again, this classification is useful. By knowing if a virus that has a single stranded RNA genome has a positive or negative genome, we get information about it. So by knowing whether the genome is positive or negative, we're understanding how it replicates. Very valuable information. Okay, the last classification criteria is whether the virus is enveloped or unenveloped. So we said we can essentially classify all viruses based on whether or not they have an envelope. So any virus, we can take it and we can say there is a virus, there is an envelope, or there isn't an envelope. So it's a good classification. It's comprehensive, and it matters because, as we'll discuss, it impacts how the virus replicates. You'll see it's the bottom classification line here. So we put the virus through all the other classification systems. Then right at the end, we just say, okay, does it have an envelope or does it not have an envelope? In terms of how this envelope, if it's present, impacts how the virus replicates, we have to think about where the envelope comes from. And this is key. The envelope is derived from the host cell membrane. So the envelope is actually a small portion of the host cell membrane that the virus steals when it leaves an infected cell. When it enters into a new cell, usually the viral envelope then fuses with the host cell membrane. So it's essentially using it as a bit of a transport vessel to get from cell to cell. It gives it a bit of protection, embeds some certain proteins in the envelope sometimes that helps it attach to new cells. Obviously, because you have this process of budding where the virus steals a bit of the envelope when it leaves a cell, and then you have the process of fusion where the viral envelope fuses with the cell membrane when the virus enters a new cell. These are specific events that only occur in viruses that have an envelope. You may have heard of drugs called fusion inhibitors. Fusion inhibitors are antivirals that inhibit the process of fusion. So they prevent enveloped viruses from fusing their envelope with the host cell membrane, and it stops these viruses from entering into the cell. Very effective antivirals, but obviously these drugs only work against viruses with envelopes. So again, clear example of why this classification system works, or one of the ways in which it's useful. So I went into some detail kind of defining why we have these issues phylogenetically classifying viruses, and everything I said was accurate, I hope. Yeah, everything I said is fine and true. However, phylogenetic classification of viruses can still be useful. We don't want to get rid of the classification system completely. As you saw with the phenotypic classification system, we're still using elements of phylogenetic classification. We're still using virus families, and we're putting the virus families onto this phenotypic map. So obviously there's some value in the phylogenetic classification. So we do still use it to a degree, which is what I want to talk about now. For example, these are the issues we talked about. So we talked about the idea that you can't really classify higher than orders. We talked about the idea that we can classify into families. We can do this fairly consistently, and it works fairly well. We can group viruses into these little groups, into these families. And again, that's what we're applying to the phenotypic classification system. We're taking the families and we're putting them onto the phenotypic map. These are the orders that we currently have for viruses. This includes bacteriophages. We can roughly align these orders with phenotypic classification. So this is interesting. So when we take the orders of viruses that do exist, and bear in mind these orders are changing all the time, also bear in mind these orders are not comprehensive. As we said, most viruses don't fit into orders. Some do fit into orders, but maybe not particularly well. And of the orders that do exist, there's quite a lot of them. We don't understand how these orders are related to each other. However, if we apply these orders to the phenotypic classification system, we see something very, very interesting. And what we see is each of these orders tends to be clumped in a specific part of the phenotypic system. So what that means is each individual order has consistency within it. So all the viruses in each order have shared phenotypic features. They have phenotypic similarities. And that's what we should have expected, really, because at the end of the day, the phenotype is just an expression of the genotype. So if we're saying these orders are groups of viruses that are phylogenetically similar, so genetically they are related, which is exactly what phylogenetics is, then we should expect that they have phenotypic similarities, and that's exactly what we see. If, for example, we have a look at herpes virales, they tend to group around here. So members of the herpes virales order are DNA viruses. They have double-stranded genomes and they are enveloped. Same for mononegavirales. These are RNA viruses with negative single-stranded genomes and they have an envelope. So again, obviously it doesn't work for all the orders, but the fundamental principle is there that we do tend to see some overlap. It's not extensive, but we do tend to see overlap and consistency between phenotypic and phylogenetic classification. And the implication there is, maybe we are onto something with phylogenetic classification. Maybe we just don't have all the information we need. And there are attempts to re-envisage the phylogenetic classification system in a way that helps us to include all viruses, and we'll talk about that in a few slides. It's quite interesting. We have to remember as well that these terms, these levels that we've added, species, order, kingdom, phylum, at the end of the day, we've made them up. We've made up those terms and we've applied them to try and make sense of the natural world. In the natural world, It's just a bit, the tree of life is a big sprawling tree. There aren't these clean divisions. We like clean divisions because we're human, and it's particularly scientists. We like to have things ordered and like to have them making sense and understandable. That's not really the natural world, though. Again, there aren't clear divisions between species and families all the time. Yeah, so maybe we have to rethink a little bit how we approach phylogenetic classification of viruses. Okay, so I'm just elaborating a bit on trying to kind of take phenotypic classification and phylogenetic classification and make them work together. It kind of works. Again, as I said, we see these groupings that overlap, but they're not absolute, and this is a good example. Single-stranded negative viruses that are enveloped, it's a grouping of mononegavirals. This is where mononegavirals fit. However, not all viruses that fit the criteria, not all viruses that are single-stranded with negative sense genome are mononegavirals. It just doesn't always apply. And here we have an example of the strategies that are trying to be employed to get around the limitations with the phylogenetic classification. Again, I really want to emphasize the fact that they're just limitations of phylogenetics. It's not that phylogenetics is completely useless. It's just that it has these specific limitations that we need to mitigate or get around. And one of the ways we get around them is by using the phenotypic classification system, or at least using it alongside phylogenetic classification. This system proposes to get away from the problems associated with phylogenetics by using a different kind of step system. So the idea is the same. The overall principle of classifying viruses phylogenetically is the same. So it's based around looking at genetic similarity and grouping viruses that are similar together. However, instead of the traditional rank structure of species, genus, subfamily, family, order, and then not really much further, it proposes using a slightly different structure that, according to the researchers who've put this forth, allows you to classify all the way up to realm, so allows you to get this full classification system. You can see on the left that, as I've been saying, the classification of viruses in the traditional phylogenetic system doesn't go past order, so the limitations just don't allow you to group these viruses together further than order, so any higher up than order. This classification system, however, apparently allows it all the way up to realm. It's interesting. What's interesting about it, apart from it being inherently new and kind of cool, is that this was published in Nature and it's been taken quite seriously. Quite recently, 2019 is when it was published. However, it's had quite an impact, and we do see these shifts occasionally in science where there's just a big change. It's decided that, oh, things are going to be done differently now. We're going to use this terminology, or we're going to use this kind of abbreviation system. And it's possible that there could be a big jump from kind of struggling by with the 5-rank system that we have been doing, and instead using this 15-rank structure. Just something to think about. Again, it is quite new, but it will be interesting to see if it's adopted. And here are some of the, well, it's basically some quotes from the paper that I've put this forward. They're outlining the issues with phylogenetics as we have, and they're putting forward their argument as to why this new system should be used. And you do see this a lot in scientific papers. When a new way of doing things is put forward, it has to be justified. If you're interested in this kind of thing, I would encourage you to read that paper, by the way. Is it? I haven't linked it. I'll link that. I'll put it on the module space. I'll make a note of that actually. Module space. Okay, sorry. Okay, so that's phylogenetic classification. Again, limited but still useful. Baltimore classification system next. Okay, and last of all, I want to talk about the Baltimore classification system. Again, you'll see this classification system is really closely tied to the other classification systems. And hopefully you'll understand what I mean by that as I explain the details of it. So again, just a reminder that when we're using the phylogenetic classification system, we're not able to go any higher than order, so we just really get stuck at order. We just can't go any further. So that's the necessity for these other classification systems. And if you actually look at the Wikipedia pages for these different viruses, you'll see they're classified up to order, as we talked about, using phylogenetic classification. However, above order, there's something else, something unusual. There's something called group. Here we have for HIV group 6, then group 5 for Ebola virus, then group 7 for hepatitis B virus. So what does this mean? It's not phylogenetic, because if we look at the phylogenetic classification system, it's not there. It's not one of the kind of levels of classification that's used. So it must be something else. And the something else that it is a classification within the Baltimore system. So it's important, and it's important enough to be present on the Wikipedia page. Obviously, there's issues with Wikipedia, but if nothing else, it being included in the core information of these important viruses on Wikipedia is at least evidence that it's widely used in some circles. And again, just highlighting that these specific groups are not phylogenetic. The term group is referring to some other type of classification. And as we're going to talk about, the specific classification system that group is referring to is the Baltimore classification system. I highlight here that it doesn't particularly make a lot of sense to have the group at the top here, because it kind of implies that it's the next level up from order. In phylogenetic classification, you have the genus and family and order. Then this group, again, just because the way it's laid out, it implies group is a level above order in the phylogenetic classification, but it's not. It's a completely different classification system, so we don't want to get confused between those. So just a bit more information on the Baltimore classification. Baltimore classification essentially splits viruses into a series of groups, so a number of different groups, and these groups are based upon the way each virus transcribes and translates its RNA. So the way it expresses its genome, I shouldn't say RNA, I should say genome, because some viruses have DNA genomes. So the way it expresses its genome, and also the way it replicates its genome. Obviously, the way viruses transcribe and translate, so the way they express their genome, and also the way they replicate their genome, is highly dependent upon the type of genome they have. So whether or not they have a DNA genome or an RNA genome or a single-stranded genome. Therefore, the Baltimore classification system overlaps very significantly with the phenotypic classification system, because these are features we use in phenotypic classification to classify the viruses. If a virus is a DNA virus, if it has a DNA genome, it goes on this part of the classification, that phenotypic map we looked at. If it has an RNA genome, it goes on this part of the phenotypic map. If it has a single-stranded genome, it goes on this part. So these are all features to do with the genome, and they all impact the Baltimore classification. So that means there's this high level of consistency between Baltimore classification and phenotypic classification. This is the simplest map you'll see of the Baltimore classification. It does look complicated, and it is a little bit complicated. I'm going to explain it as best as I can. We'll go through it as well in the tutorial. It's a bit tricky to explain one way. It doesn't lend itself well to a simple didactic explanation. It really needs to be explored. So we will focus on this in the tutorial, but I can at least hopefully clearly describe what it is, and then we can explore that description in the tutorial. So at the bottom, we have mRNA. These arrows represent expression. So what is essentially happening is we're going from the viral genome to mRNA, and the part of the Baltimore classification is how is the virus getting from its own genome to mRNA. The reason why I say genome replication is included in the Baltimore classification is because some of these viruses have mRNA genomes. Excuse me. They have mRNA genomes, so their genomes are composed of positive stranded RNA, so it's RNA that can be turned straight into protein. So for them, genome expression is exactly the same as genome replication. They're making the same thing. They're making positive stranded RNA. So that's one key takeaway, that all of these viruses are going to mRNA. That's where the arrows are going. That's because all these viruses, when they infect a cell, they're using the cell translation machinery, and the cell translation machinery, it uses mRNA to create protein. It needs an mRNA template. So for any virus to produce proteins, it has to do this via mRNA. Again, we'll talk about this in detail in later lectures, but that's a really key thing there. The top of the classification, you'll see that for group 1, we have dsDNA, double-stranded DNA. For group 2, single-stranded DNA. Group 3 is double-stranded RNA. Group 4 and 5 are positive and negative stranded single-stranded RNA, respectively. Group 6 and Group 7 are slightly different, but for the first 5 groups, you can see the grouping is based upon what their genome is made of, and then you have this arrow from what their genome is made of going to RNA. And the reason why these groups exist as they do, because the way these viruses produce RNA, so the way they express their genome, is entirely dependent upon what their genome is composed of. So the double-stranded DNA viruses, they all create RNA the same way, so they get put together. The single-stranded DNA viruses, they all create mRNA the same way, so they get put together. It's the same for each of these 5 groupings. You see this light green box as well on some of the arrows. This is an intermediate. Again, we'll go into this in a little bit more detail, but it essentially means that group 2 viruses, they get to RNA through a double-stranded DNA intermediate. Group 4 viruses get to mRNA through a negative single-stranded RNA intermediate. We have specific lectures where we look at genome replication and genome expression. It's beyond the scope of this lecture to look at it in any more detail than we're looking at it right now. So please don't worry that there's big gaps missing in what we're talking about now. We're going to fill in these gaps later. Specifically the gaps around group 6 and group 7. All we need to know is that at this stage is that these viruses are grouped separately because they use reverse transcriptase. Reverse transcriptase is a very, very specific enzyme that does a very, very specific thing, and it basically allows viruses in these groups to express their genome, so to produce RNA in a way that is completely different to the way other viruses are doing it. Same for genome replication. The way these viruses replicate their genome, so make copies of their genome, is completely different to the other viruses, just because there's reverse transcriptase involved. So again, a little bit vague, but hopefully I'm explaining the principles of how the classification works. Again, what we see when we line up this classification with the phenotypic system, as I kind of explained earlier, we see this huge overlap because the way the genomes are expressed and the way the genomes are replicated is almost entirely dependent upon what the genomes are composed of. And this phenotypic classification is also almost entirely dependent upon what the viral genomes are composed of. So we see group 1 viruses, so group 1 of the Baltimore system viruses. These are double-stranded DNA viruses, and they're grouped together because they have double-stranded DNA, so they all express their genome the same way. They all replicate their genome the same way. Exactly the same thing going on. When we compare this to the phenotypic system, we see double-stranded DNA viruses are also grouped together here, which again completely makes sense with what we understand about the system. Same for group 2, or sorry, I'm calling with the Baltimore system, sometimes they'll be referred to as groups, sometimes they'll be referred to as classes, exactly the same thing. So with group 2, we have single-stranded DNA viruses. Again, all these single-stranded DNA viruses will essentially replicate and express their genome the same way. Same for group 3, same for group 4, same for group 5. So again, this Baltimore classification system is completely consistent with phenotypic classification because both are essentially fundamentally dependent upon the composition of the viral genome. Group 6 and group 7, sorry, I cut myself off there. Group 6 and group 7 are slightly different, again, because they have reverse transcriptase. So we're going to consider those separately in later lectures. All we need to know at this point is the first 5 classes of the Baltimore classification system are consistent with phenotypic classification, and we need to understand why that's the case. Hopefully we do. The latter 2 groups, again, we're going to consider those later when we delve into the details of genome expression and replication. And once we've done that, we should have a really solid understanding of why those groups are separate to the other groups. And again, all revolves around reverse transcriptase, which is an enzyme, and the process that enzyme mediates, which is reverse transcription. Okay, just a couple of examples here. Ebola virus, again, we can see group 5, negative single-stranded RNA. So this is where it is. In group 5, it's an RNA virus, so it's down in the bottom half of the diagram in the middle. Group 4, we have hepatitis C, so positive single-stranded RNA virus. Again, same, just to the left of class 5, so it's in the bottom left of the RNA virus map. So this Baltimore classification is really very consistent with the phenotypic classification system. Okay, so I hope that's been clear. Again, if anything isn't clear, we're going to go through it in the tutorial. Again, I've kind of whizzed over a couple of specific things, so it might feel like there's some gaps, but I've done that deliberately. The reason is it's just beyond the scope of this lecture. These are quite complex processes, things like genome expression and replication, and they each deserve a full lecture so we can really fully explore them, fully understand them. They do completely make sense once you've put that time in, once you've spent a full lecture pulling them apart, understanding what's what, really understanding the definitions. So don't worry if anything seems a bit complicated now. At this level, we're just trying to establish the fundamental principles, and then we'll delve into the nitty-gritty in the next few lectures. Okay, thank you very much.